back to the gym yesterday. I think I worked out for 20 minutes. Yeah, COVID really, I mean, I've been working out since 1989, and I never stopped for more than a few weeks, and that all the way up until 2020. And then with COVID, you know, I don't live in town, and we weren't coming to class anymore, so it was a long time off. Man. And it was kind of cool. I just read today in CNN, there was this really large study that came out, another study just showing the benefits of exercise. And in this case, I don't see anybody in here over the age of 40, but you will be one day. You're going to look around and go, what the hell just happened to me? But even though at the age of over 40, they're saying just 10 minutes of a brisk walk every day is enough to really make a difference in your life. And if you walk more, even more. So I was really happy to be back. I've been hiking the whole time. I hiked 600 miles last year. Yeah, I'm an avid hiker. That doesn't show it, but okay. What I'm going to do today is quickly review some just basic chemistry, so we're all on the same page here. And then I'm going to review just briefly eukaryotic cells, and then I'm going to start histology, because as I was going through the notes and reading the chapter, I realized that just some basic knowledge or refresher might be helpful. Okay, so this is in the first two chapters, chapters 2 and 3 of your A&P book, but I'm going to do them here in about 20 minutes. I won't go too fast. It's just I don't want to cover all of it, just very basic stuff. Okay, in the chemistry part, you guys remember covalent bonds, hydrogen bonds, polar, nonpolar covalent bonds. Who wants a refresher on that? Okay, really quickly, if we have a molecule like can you guys see all right? Can you see all right? Yes, no? Okay, cool. So I have a molecule like methane, which is carbon surrounded by 4 hydrogens. The chemical formula, let's see if I can find some new pens. The chemical formula is CH4. This means I've got 1 carbon, 4 hydrogens, and they're held together by these chemical bonds. And these particular chemical bonds are called a covalent bond. And a covalent bond basically means I'm sharing my outermost electrons. So if you've got an atom, you've got the nucleus in the center with your protons and neutrons, and then orbiting that, and your electron shells are your electrons, and your outermost shell are those valence electrons. And if this was carbon, here's hydrogen, and you share those electrons, forming a covalent bond because you're sharing your valence electrons. Good? Everybody got that? And of course, these electrons are shared equally. Be a non-polar covalent bond. And then if I have something like this molecule right here, very common one on this planet, is water. Water is, of course, made with oxygen and hydrogen. This bond right here is a polar covalent bond. And the reason why that the electrons are not shared equally, because oxygen is more electronegative than hydrogen. Electronegativity is a measure of how much an atom attracts electrons. Electrons are negatively charged, protons are positively charged. Opposite charges attract, right? So oxygen has 8 protons to its 1, so there's 8 protons pull these electrons to it, and it becomes a polar molecule. And of course, these are delta symbols, which mean partial negative charge, partial positive charge. Okay, everybody good here? And this is a polar molecule because it's got a partial negative end and a partial positive end. And of course, polar molecules are hydrophilic. They dissolve in water. Water attracts to it, is attached to it, or attracts itself. That would be a hydrogen bond. And that is why water is liquid, because the water molecules stick to each other because of this attraction here. Okay? Is that helpful? Yeah, I mean, just because you learned it once doesn't mean you remember it or you remember it well. So I don't have a problem going over stuff you've learned in the past. A lot of other places, they assume you know it, and then they move on, and that can be tricky for people. Okay, now, so of course, those were molecules, and there are 4 major classes of molecules for life called the macromolecules. Do you guys remember the macromolecules? Yeah.
Proteins, lipids, carbs, and
Doing good, man. It's been a couple years since you were in my class, so. The nucleic acids. Okay, not every organic molecule, and of course organic molecules include carbon and at least 1 hydrogen. Okay, so organic molecules come in these 4 classes, but not every organic molecule fits easily into one of these 4 classes. So like vitamin D, vitamin K, vitamin A, beta carotene, vitamin C. These are organic molecules that don't fit nicely in here, some of your vitamins. The vast majority of organic molecules do fit into one of these groups, specifically this one right here. So the macromolecules include proteins. And if you remember, proteins are made up of building blocks called amino acids. That's not a 6, sorry about that. These are, of course, amino acids. There are roughly 20 or so amino acids used by all life to make hundreds of thousands, perhaps millions of different types of proteins. Okay, so these are the building blocks. This R is not an element. This R represents a functional group. So you have approximately 20 different functional groups, the simplest one being a hydrogen. They could be a hydroxyl group. They could be as simple as a methyl group. You get the idea. There's like 20 of these different things, and there are these different functional groups that make up the 20 amino acids that make up the vast majority of proteins. Okay? So to me, it's always like proteins are a lot like Legos. I always want the Lego Millennium Falcon. I've been waiting for my 4-year-old to ask for it. She plays lightsabers with me, but I'm really waiting for her to ask for the Lego Millennium Falcon. And here's why. If this is your Lego piece, you can take these pieces and build up different types of Lego spaceships from Star Wars, like an X-Wing or a TIE Fighter or a Millennium Falcon. And if you don't know Star Wars, Harry Potter's castle, Hogwarts, I don't know, right? You get the idea. You have these building blocks, and with these 20 different building blocks, depending on how many you use and fold them, you can create almost an infinite number of Lego things, right? I always play spaceships because I love sci-fi. Or you can build up almost an infinite number of proteins. Okay? So proteins are what we call a polymer made up of the building blocks amino acids. And of course, the primary structure of a protein is just your sequence of amino acids. And then proteins, of course, have primary, secondary, tertiary structure. For us, the tertiary structure of a protein is what it's folded. When a protein gets folded into its shape and becomes biologically active. And you can also, of course, have a 4th level of structure called quaternary structure, where you've got multiple proteins, or what we call peptides, coming together to form a single protein. So your hemoglobin in your blood has quaternary structure because it's made up of 4 peptides Peptides are nothing more than a whole bunch of amino acids strung together, right? And they all fold together to form a protein. So importantly, proteins are the workhorses of our cells, of life. Your muscles, made of proteins, right? Your collagen that we're going to learn today is made of proteins. So much of what we're going to learn about histology and the extracellular matrix are proteins. Gap junctions, desmosomes, tight junctions that we're going to learn about today. Those are proteins. Proteins also form enzymes. Lactase. How many of you can drink milk? Yeah, you mutants. You're making lactase as an adult. No other mammal in the history of mammals has ever been able to milk as an adult because they don't produce lactase as an adult. Your switch broke. You don't turn it off as an adult. That means I suffer when I eat ice cream and I'm just really jealous. But the point is that lactase is an enzyme. So proteins, are muscles, collagen. They form, the proteins that hold our cells together, protein channels. They also are enzymes that speed up chemical reactions. You get the idea. There are proteins in your eye that allow you to see light. Everybody good with proteins? So whenever you eat a steak, you break your proteins down into your amino. Since we mentioned lipids second, we'll go ahead and do lipids second. Lipids, what are they? Fats, waxes, oils. So your olive oil is a lipid. Lipids are typically pure water. That is why oil, your olive oil, like an olive oil balsamic vinaigrette makes a great salad dressing. But your olive oil always flows to the top. That is because olive oil is hydrophobic. It's mostly carbon and hydrogen. It cannot form hydrogen bonds with water, so it separates from water. Okay. Also importantly, not only do they form fats, as we're going to learn, in adipose tissue. I got some right here, the love handles, right? They tend to grow up on you. You got to watch out. But that's from adipose tissue storing fats, which are a type of lipid called a triglyceride. When you go get your blood levels checked, your blood chemistry checked, you want low levels of triglycerides because triglycerides are blood fat, and you don't want that because they form plaque on your arteries. Okay? You guys know what a triglyceride is? Anybody not know? Okay, cool. You have a glycerol, which is this 3-carbon thing here, attached to a fatty acid. And because there are 3 of them, hence the name tri, because there's 3 of these fatty acids, glycerol, triglyceride. This is fat. Okay, specifically, this would be saturated fat. This is, of course, your carboxyl group. This is your carbon. I didn't draw the hydrogens on here because I'm a little lazy. This line right here, what does that represent? A covalent bond. This point right here represents a carbon, and there would be 2 hydrogens. So at each of these nodes, there's a carbon and a hydrogen. Of course, this thing is hydrophobic, and fats store lots of energy in these covalent bonds. So fats store lots of energy, much more than carbohydrates, okay? And then, of course, when we start talking about our cells are all surrounded by a membrane, a phospholipid bilayer. This would be a phosphate group. This is a lipid, hence the name phospholipid. And I'm making a point of this because we're going to see all of these names like glycosamine, right? Glycosamine, amino acid, glucose, sugar, and peptides, right? Sugar and amino acids. Okay, so this would be a phospholipid. And of course, you know that all of your cellular membranes are made up of a phospholipid bilayer, which we would draw something that looks like this. So when we start talking about cells going, or transcellular transport is going through a cell, you have to remember about transport. Is it going through there through diffusion, facilitated diffusion, and active transport? So this is what surrounds every single membrane, cellular, at the cellular level, not the basal lamina or the basal membranes in our tissues, but at the cellular level. These two tails, what do they represent? Fatty acid, exactly. And these two tails are hydrophobic. What does this round ball represent? Phosphorus. And so this is hydrophilic, right? So this thing is amphipathic. It's got a hydrophilic region, a hydrophobic region. And because this region in here is hydrophobic, it has selective permeability. Okay, so all your cellular membranes are surrounded by a plasma membrane, also known as a phospholipid bilayer. Phospho is a lipid, there's two layers, bilayer. So we got to start breaking down our words. It helps us to look at the words and the roots, and that way we can get at what their understanding of them. Okay, any questions? Anybody not understand? All right, lipids also include your hormones like testosterone, estrogens, estradiols, plant sterols, and also cholesterol. Okay, all right, any questions here? And then carbohydrates. Carbo means, take a wild shot at this one. Carbon and hydrates, which means it's got hydroxyl groups on it. So our famous molecule here, which molecule is this? It could be glucose, or it could be fructose or galactose, but it's typically we call it glucose, right? And glucose, of course, I'm still getting back in the swing of lecturing after 2 years. But at any rate, molecule hydrophobic or hydrophilic? Hydrophilic. Why? The OH group, it can form hydrogen bonds with water. That's why you can have a Mountain Dew that's 12 ounces and like 40 grams of sugar, right? Yeah, I go for the diet stuff. I take all the aspartame and sucralose. Yeah. Okay, any questions here with carbs? This would be a simple carbohydrate. Okay. Now, if you remember cellulose, glycogen, what do you know about those molecules? Simple or complex? Starch, like rice, potato. Yes, did you have a question? Or? Oh, yeah. Oh, okay. No worries. Sorry about that. I saw the hand go up, so I was like, oh. Yeah, so simple carbs are sugars like glucose, fructose. Fructose, of course, is an isomer. That looks like this. It's got a 5-membered ring versus a 6-membered ring. So that's the difference. That's why it's an isomer. Same chemical formula, C6H12O6, with different structural formula. Instead of a 6-membered ring, as a 5-membered ring. But these are simple sugars, simple carbs. Okay, complex carbohydrates are you take a whole bunch of glucose molecules and you string them together, and you get cellulose, you get starch, which is like a potato, and then you get glycogen, which is a complex carbohydrate stored in your liver and in your muscles. Okay, any questions? All right, is this helpful or? Okay, good. So a lot of times if you see the term saccharin, saccharide, that means sugar. Glyco, glucose. Glyco means sugar. So if you have a glycoamine or a glycopeptide, peptides are a string of amino acids. So you can put sugars and attach them to proteins or to small strings of amino acids. Or you can add an amino acid to a sugar, right? And you get a glycosamine, right? So we have glycopeptides, that's where this comes into play. You can also attach carbohydrates to lipids, and then you get a sugar lipid, a glycolipid, okay? So we talk about these 4 classes of macromolecules, but these 3, you can stick amino acids to carbohydrates, small short chains of carbohydrates to proteins. And in fact, you ever heard of A, B, and O blood? A and B blood are different branching types of carbohydrates attached to a protein on the surface of your blood cells. If you're O blood, it's a truncated A version, it's a mutated A. You don't produce the A antigen. Okay, nucleic acids, you guys remember, not so important in here, but just though we talk about them, DNA and RNA, life stores information in our DNA. RNA is used to actually go out into the cell and carry out many of the cellular functions directed by DNA. So DNA doesn't make proteins. It stores information to make those proteins and regulate the cell's activity. You copy RNA from your DNA. Your RNA goes out into the cell, either makes proteins or regulates their activity. Okay? Any questions on any of this? Okay, so just wanted to make sure we were all on the same page, so that way when we start saying terms like collagen and it says it's a protein, you know what it is. Or if we talk about a peptidoglycan, pepto has to do with a string of amino acids, peptides. All proteins are peptides. Every single protein is a peptide because a peptide is a bunch of amino acids strung together. Peptides can be folded into proteins. We usually say that proteins have biological action, whereas a peptide is just a string of amino acids. Okay?
Peptides make up polymers, or?
Peptide is a polymer. So anytime you have like a bunch of amino acids, the amino acids are the monomer. Mono means one, right? So if I have a polymer, that means I have many amino acids. So DNA, RNA are polymers made up of building blocks. Polymer, polymer, polymer. The building block is amino acid. The building block of complex carbohydrates is glucose. The building block of nucleic acids is either DNA or RNA, which is adenine, thymine, guanine, and cytosine. And if you're using RNA, you have uracil instead. And you're using a ribose sugar instead of a deoxyribose sugar, which those differences aren't that important here. If somebody wants to know, I can tell you. But missing the hydroxyl group on number 3. So there you go. Okay, anybody else? We good? All right. I noticed also in the histology, like I said, we went right over the cell biology part. I'm also teaching cell and molecular biology, so we haven't got to studying cells yet. But ironically, as somebody who likes birds and lizards and fish and ecology and natural history, ended up really liking cell and molecular biology. And in this class, it's not that important, but I feel like it's helpful to be on the same page with our cellular biology, because once again, even in histology, they were talking about cellular parts, and it is helpful to know it. So how well do you guys remember your cell biology? Pretty well? Yeah. First time I learned it took me a while. So of course, you and I are made up of trillions of eukaryotic cells. What makes a eukaryotic cell different from a prokaryotic cell, like a bacteria? Nucleus. Anything else? What's that?
Organelles.
Organelles, mitochondria. In reality, from an evolutionary point of view, the defining character of eukaryotes is actually the mitochondria, not the cell nucleus. The cell nucleus most likely evolved in response to the mitochondria. Interesting, isn't it? But when we started studying cells hundreds of years ago, when they invented the first microscopes, they noticed you were either bacteria, prokaryote, you lacked a nucleus, or you had a nucleus. So, here we are still teaching that eukaryotes are defined by the nucleus. And that's not a bad way to define them, because all eukaryotes have a cell nucleus. And the cell nucleus is defined as having a nuclear envelope right through there, okay? And inside of it is this structure of the stuff called chromatin, which is your DNA plus a whole bunch of proteins. There are lots of proteins inside the cell nucleus along with your DNA. There's some really cool stuff I just love to share here. Inside of each one of your cells, you have 3 billion base pairs of DNA. If I were to take that out, the DNA of one of your cells and take it out, it would be 6 feet long. Could you imagine like having a ball of yarn that stretches from here to Denver and you wrap it up and put it in this room? Not only that, the chromatin is highly organized, and is organized depending on which type of cell it is. So we have 200 different types of cells that go on and form, our tissues. We're going to learn about 4 different tissues. Tissues go on to form organ systems, right? Organs and organ systems. So depending if you're a nerve cell or a muscle cell, this chromatin is organized differently, or a skin cell. And your skin cells that divide all the time will condense this into visible chromosomes, divide through mitosis, and then the chromatin will come back out. So imagine a ball of yarn that stretches from here to Denver. We wrap it up and put it in this room with all these scaffolding proteins. And over in this corner is always the gene to make, to make lactase. And in your stomach cell, or in your intestines where you need lactase, it might be over here and all spread out. But in your muscle cell, you'll never see lactose in your muscle cell, milk sugar in your muscle cell. It might be just tied up and packed away up there, like your Christmas gifts. Or Christmas ornaments, I should say. That makes sense? It's pretty wild, isn't it? Just the level of organization is just incredible here. The nuclear envelope, of course, is a double membrane. Think of like a bunch of sandwich bags all stitched together so the whole thing comes apart. And then you have your energy converting organelle called the mitochondria. And in a lot of textbooks, you hear powerhouse of the cell. You see a diagram where there's 3 or 4 mitochondria in the cell. Well, mitochondria are much more important than simply making ATP. Now, of course, I shouldn't say simply. You make your body weight like every day in ATP. I'm gonna lie about my weight. I make 180 pounds of ATP every day. I actually make a little more than that, but I'm lying. I'm fibbing. I need to find me a new scale. But mitochondria are also involved in the evolution of eukaryotes, like the evolution of 2 sexes, sexual reproduction, programmed cell death. Every day you're losing like 50 billion cells because of mitochondria involved with programmed cell death. And of course, they make ATP. And they're also the defining character from an evolutionary standpoint of eukaryotes. Good? Yeah, it's my favorite organelle. It's such a cool little thing. The other thing I was going to say about the diagrams in most textbooks, they show 3 or 4 mitochondria. No. most of your cells has tens to hundreds metabolically active cells, like the cells lining your stomach that make hydrochloric acid that pump hydrogen protons into your stomach. They're loaded with mitochondria, thousands of mitochondria, because they're actively lowering the pH in your stomach. Anybody in here an athlete, a runner, a swimmer, long distance stuff? Yeah. Your muscle cells have thousands of mitochondria in them. And if you become more active, you'll actually get more mitochondria. Exercise is crucial to our health and well-being. Okay. We also have what's called the Endomembrane System. And it's a child of I grew up in the 80s. I was born in the 70s. I cannot, every time I hear it, and now my daughter jumps around to it. It's awesome. Okay, the endomembrane system, very quickly, that are involved with the synthesis and breakdown of cellular components. Okay, so the endomembrane system. So you have what's called the endoplasmic reticulum, and you have both smooth, including the sarcoplasmic, which we're going to study in this class, which is a smooth type of endoplasmic reticulum in your muscle cells. It's used to store ions, specifically calcium ions, because you have to have calcium ions for your muscles to contract. And you've got the rough ER, and the rough ER has ribosomes on it. And I forgot to mention ribosomes really quick. Ribosomes are found in every single cell on this planet. And what's special about a ribosome? What do they do? Exactly, protein synthesis. Synthesize means putting together. Every single protein on this planet was made inside of a ribosome inside a cell. Okay? Every single protein. Any protein that is going to be exported outside the cell. And today we're going to talk about the extracellular matrix. All these different proteins like collagen, they are outside of cells. But the proteins are made inside, they are assembled inside the cell. And then they are exported outside of the cell. And they're the translation of the amino acids into, or the translation of messenger RNA into a sequence of amino acids to become like collagen takes place inside the rough ER. So we've got, this is a side of protein synthesis. So the ribosome is putting together the protein into the lumen of the rough ER. We also have these various transport vesicles. So if I had the rough ER here, these little dots right there represent the ribosomes making the proteins. It's called rough ER because, well, it's got ribosomes on it and it's rough, not smooth. What will happen is you'll make the proteins inside the lumen of the rough ER, and then you'll have a transport vesicle will bud off and travel to this series of sacs called the Golgi Body or Golgi Apparatus, but embarrassingly, because I type on Microsoft Office all the time, I can never remember how to spell apparatus, so I just say Golgi Body, which is totally acceptable. Yeah, man, you forget things when you type on computer for 25 years, right? I don't have that stored in my memory anymore. How to spell apparatus, simple words just elude me. So at any rate, the Golgi Body will take the protein from, you'll assemble it here. This is like, you're at Ford, you're making your F-250, your 3/4 ton truck here. You get it mostly done, you stick it in the transport vesicle, take it to the Golgi Body. Here you're gonna put the finishing touches on it. Put the paint job on it, put the carpet in there, stick the radio in there, spray the new car smell in it. No, seriously, it's a smell. You can actually get new car smell for your car. I'm not kidding. It's a thing you can do. At any rate, The Golgi body is where you would take the protein, finish its folding, and potentially activate it, put it in another transport vesicle where it will leave the trans side of your Golgi body, come out here to the membrane, and be released outside the cell. And it could be a protein for like amylase. Amylase is in your saliva. It helps you break down carbohydrates when you're eating bread. It could be the proteins used to form collagen for the extracellular matrix. It could be a number of things. We good? Yeah.
Is it removed by exocytosis or something else?
Yeah, that's part of exocytosis. It means going outside the cell, because this transport vesicle is budding. It's attaching to it, and they're like soap bubbles, right? Similar to soap bubbles. You ever seen soap bubbles? They'll come and stick together, and then they'll merge. That's more or less what's happening here. And that's also a way for the cell to create new membrane and to get membranes embedded into the proteins embedded in this membrane as well. And we'll see this when we look at secretory vesicles in the epithelial cells, right? We have vesicles, we have cells that actually release oils, they release hormones, they release things, and they're doing, this is one of the ways they do that. Okay, any questions here? Okay, one last thing. We also have what is called, this is most of the endomembrane system. You know, these set of connected and interconnected organelles that include the endoplasmic reticulum, smooth and rough, Golgi bodies, various transport vesicles. I'm not going to go into this too much here. Lysosomes. Lysosomes are incredibly important for breaking down things and recycling them. And for anybody interested in aging, they're very important in the aging process in terms of autophagy. Auto means self, phagy means eating. You're seeing little kids. My 4-year-old runs all over the place. She's got tons of energy. My dad can barely walk from here to the end of the building. He's like almost 80. And the difference is here, as we get older, we lose energy. One of the reasons why we think we lose energy as we age is because the lysosomes slowed down and the autophagy of mitochondria. So what happens is, as we age, we start accumulating less efficient mitochondria. Mitochondria make ATP. If we have less ATP, we have less energy, we slow down. Pretty wild, isn't it? Yeah. Turns out we can reset that, which is kind of cool. Okay. There's other cellular structures like peroxisomes, not going to talk about that here. And then we have the cytoskeleton. A lot when we get to muscles, because your muscles are actin and myosin. So there are two different proteins working together to help the filaments slide past each other. Today we're going to talk about keratin fibers. This is a class of very strong proteins that forms like our nails, parts of our hair, and then our skin. They're very strong. And then, of course, the microtubules. These things are very important. It's all very important. Every one of them has their functions. Microtubules are like hollow tubes, and they're modular. The cell can build them up and take them down. Microtubules form things like the flagella. So a lot of male infertility has to do with the flagella not working because the microtubule is not working correctly. We have cilia in our respiratory tract. You start getting a little sick, you start kicking out all that mucus. That's cilia trying to beat that out of us, right? And it protects us. Cilia are also made up of microtubules. The entire architecture of this cell, when these transport vesicles, they're not just floating from here to here or from a Golgi body to the cell membrane, the outer membrane. They're being transported by motor proteins attached to the transport vesicle, walking it down the microtubule. So there are microtubules throughout our cells involved with moving all of these transport vesicles around, and they also help anchor some of the organs in place And then in some cases, using actin filaments and microtubules, you can actually move the organs around with motor proteins. And it's also important for giving the cell its shape and allowing things like our macrophages, which are our big eaters, part of your immune system, to take out foreign bodies in our body, like pathogens or cells that are infected with small viruses. Okay, was that helpful? Okay, any questions about the computer here? Well. Okay, let's see here. The lights. Okay, now that we've had our little background there on chemistry and cell biology, and there's no other questions, let's begin with some histology. And I'm going to basically follow the book really closely from here on out. And like I said, this was what I just did was a rehash of chapters 2 and 3. Okay. So with histology, let's see here. What's it going to do to me here? No. This is the study of tissue. So we're on chapter 4, number 1. So histology, ology just means study of, and this is a study of tissues. And I told you that we have approximately 200 tissues in the body. All right, sorry, 200 cells. I'm sorry, 200 cells. And those cells go on to form different types of tissues. And there's about 4 different ones. And tissues are made up of cells that are structurally and functionally similar, okay? And they also have a surrounding material called the extracellular matrix. Okay. So we're going to see some abbreviations here. So ECM is the extracellular matrix. Here are the 4 basic types of tissues. Epithelial tissues, and that's what we're going to talk about today. We also have connective tissue, which include tendons, ligaments, blood. I know, it's weird to think of blood as connective tissue, but your tendons and ligaments. Ligament tear is bad. Why it takes long muscle. Your esophagus is a smooth muscle, which is moving stuff down to your stomach. And guys, we don't. I mean, it pushes a baby outside. And then, of course, there are the nervous system, which is your PNS, and all the different types of nerves. We're going to me are really exciting to know how they work. Okay, so we have these 4 basic types of tissues, and the tissues are made up of different types of cells. Now, these cells can either be touching each other, or they might not be. There might be some space between there, which is the extracellular matrix. And your extracellular matrix has a ground substance, which could be a number of things. It's water, but it could be other things in there as well, like ions and sugars and proteins. And there are also these protein fibers. And there are these different types of protein fibers outside of the cells that really hold us together. I mean, we are really held together by collagen. And it's interesting that collagen is unique to animals. It's a unique feature of all animals. No other life form on this planet forms collagen. And you need oxygen to make it too. Okay. Oh. And part of the extracellular matrix, it provides like support for us. And one of the things you can tell it's doing is it's holding everything in place. Your organs don't move around. your blood vessels don't move around that much. And it also some types of the some types of the ECM can resist stretching and compression. And then in other types, it's elastic, like your skin. You can yank on it. For transport, like both facilitated transport and active transport. So if you guys remember a sodium potassium pump, did you get that in 1140? Yeah, good. Sodium potassium pump, that's an active transport. Aquaporin would be a type of facilitated transport. And then they can also help anchor parts of your ECM, the protein fibers out there. And you'll start noticing things like glycoprotein. Well, what's a glycoprotein? Just sugar in a protein. Why would you attach a sugar to a protein? There's a couple different reasons that come to mind. Okay, we're holding ourselves together. Anybody ever gotten like jelly on their fingers? Sticky, isn't it? Carbohydrates are sticky, right? They're hydrophilic and they're sticky. So if I have a protein and I've got glycoproteins attached to them, it makes things sticky, like a gel. So the extracellular matrix is often gel-like with water. Okay, and then of course other reasons why you might have carbohydrates attached to proteins, and we're not going to talk about that here, but you'll get it in AMP2, is that every cell has identifiers so that your immune system can recognize it. And part of those identifiers are having these short branches of carbohydrates attached to proteins. So like A, B, and O blood is that. It's a short carbohydrate attached to a protein. Okay, so you can see the ground substance of the ECM. That's mostly the extracellular fluid with, you have things like proteoglycans, more sugar proteins, right? Protein fibers like collagen, elastin, and other types of fibers. Like glycosaminoglycans, right? GAGs, I mean, these are proteins. Okay, oh gosh. I'm not gonna go into the diseases here, except if you have diseases of your fibers, of your extracellular matrix and your fibers, you can be very loose. You can have all kinds of stretchy ability, which isn't necessarily good, and you can also have weak hearts. So every now and then you'll hear about a tall basketball player dropping dead on the court from a heart attack. Sometimes it's because they do push themselves too hard. Sometimes it's because these tall basketball players might have something called Marfan syndrome. They're tall and lanky, but they don't have good connective tissue. So if they get pushed too hard, that can actually affect their heart. Okay. Well, tissues are made up of cells. So there are different ways that cells are held together. There's also the extracellular matrix where they can be held together. They can also be touching each other. And there are 3 different types of proteins involved with stitching or holding cells together. And these are kind of cool, actually. And they only talk about them for a few minutes, but they do some interesting things. One is called a tight junction. And imagine like you're sewing something together, right? And you've got all these rivets. Here's a good example. How many of you ever gotten a cut and it got a little inflamed? Yeah. How many of you have ever had to go pee, but it was really hot outside and you waited and it seemed like it was okay for another 30 minutes or so? Probably the only one here. Okay, well, tight junctions can actually like loosen up and allow things to move between. So they can go from very tight where nothing gets through, and then based on what's happening inside the body and where these things are at, these tight junctions can actually loosen up and allow and become leaky. Yeah, so blood vessels is one of the places. That's why when you get an inflammation, you can actually have fluid leak into an area where you're inflamed and your immune system gets in there and it's inflaming your nerves. That's why it gets sensitive. You also have another one called a desmosome. So these are the desmosomes right here. These are really strong proteins, and they're made up of these integral proteins that go through the membrane and through the membranes of both. And if you notice, it looks like there's little hairs coming off of it. Those are actually protein fibers. They're keratin fibers that attach to the site adjacent cells. So now we're like, desmosomes are really strong, and these, what they call intermediate filaments, are keratin fibers. Keratin fibers are intermediate filaments are keratin fibers. They get their name intermediate because they're in between the size of the smallest ones, the actin filaments, and the largest ones, which are the microtubules. So in between are the intermediate, right? They're called keratin fibers because keratin is a class of proteins that make them up. So desmosomes are holding these cells together, and they're also attached to the cytokeratin, specifically the keratin fibers, that make them even stronger. And what they do is they hold the cells together wherever you have like shear forcing. So for instance, your skin. Holding your skin cells together are a lot of desmosomes. Okay. And the other thing is that they'll also allow a little bit of stuff to leak between the cells as well. The third one is a gap junction. A gap junction is right here. And what these do, these are proteins. These are all proteins. Everything is basically proteins, it seems like, doesn't it? But right here, these little gap junctions create little tunnels. And what that does is it